All right, welcome to Mr. B's Biology. We're going to be doing histology today, looking at the respiratory system, specifically the trachea, the bronchus, the bronchiole, the alveolar duct, the alveoli, and down to type 1 and type 2 alveolar cells. I want to first give credit where credit is due, so I get these images from the University of Michigan School of Medicine's website. They have a wonderful virtual microscopy that I highly recommend other people check out. I've been using them since the pandemic, especially for teaching the virtual labs that I've been teaching regarding AMP. So a wonderful resource, and I highly recommend everybody checks them out. I will definitely put their link in the show notes with this video. So the first one we're going to talk about here is going to be this trachea. And the first thing that we should talk about, obviously, is the lumen. So discovering where the lumen is when you're looking at anything, particularly that has a mucosal layer that will have a lumen, is one of the key things to identifying some of these other structures. So when I teach this to my students, I always make sure that they kind of work their way from the middle out. So the middle being the lumen. So when you can identify the lumen, then you can start to work your way through these layers of things on your way out to the outer surface of that structure. So here's the lumen here in this wonderful space. Another way to identify where the lumen is to look for the epithelial layer. So the epithelial layer tends to be thin in most of these structures, and it tends to also stain really dark. So this, of course, will be our epithelial layer here for our trachea. This is made up of the pseudostratified ciliated columnar cells, and there's also some goblet cells that'll be embedded in this layer producing mucus. And when you include those two together, you get the mucociliary escalator, that thing that is essentially trapping these particles and moving them so that they can be swallowed and destroyed or coughed out. The next layer that I want to highlight is going to be the lamina propria. So again, when we're talking about these different mucosal layers, that kind of layer that's just deep to the epithelial layer within this mucosal layer is going to be the lamina propria. So I'm going to denote that as LP. Typically, areolar connective tissue and some other things are found in this region. It can depend on, obviously, what type of structure we're talking about. Working our way down from there, I'm going to show the submucosal layer. So most of what we're seeing in this image is predominantly going to be submucosal layer. So I'm going to denote that as SM. So within the submucosal, at least of the trachea, you particularly find all these different seromucous glands, these submucosal glands involved with different things, such as secreting different types of either surfactants or antimicrobial agents, etc. The next thing that I really want to highlight is going to be we have the perichondrium right here surrounding the hyaline cartilage. All right, so that outer dark pink reddish band is going to be that perichondrium that I'm going to denote with a P, and this is all hyaline cartilage. So notice it has that very definitive characteristic of hyaline cartilage with all the different chondrocytes, and this is going to be part of that tracheal cartilage, that C-shaped tracheal cartilage ring. All right, so recall that it is a C shape. It's literally a C shape because the end is open. So it's not a fully closed tracheal ring or cartilaginous ring of this hyaline cartilage. It is C shaped. So there's an opening between the end of one of the C arms, if you will, and the other, which we can't see off image here, but if you can imagine, it's probably somewhere over here. In between all that, we have a dense muscle layer, and that is going to be your trachealis muscle. One of the great things about anatomy and physiology is it will hopefully make you a wonderful speller, or not, as in my case. So that is a trachealis muscle. All right. So we're going to work our way down now. So let's move on to the next layer here. Here I actually have one of the bronchus images in the slide. And first thing I want to highlight again is the fact that we can see hyaline cartilage. It almost looks like a C shape, but it's not. So one thing to kind of point out, one of the definitive differences between the hyaline cartilage in the trachea versus in the bronchi is that it's irregular and it's discontinuous. So we don't have this C shape. We have these large kind of structures of hyaline cartilage, but they're irregular shaped and they're also discontinuous, so they're not exactly connected one another. We can also see there's smooth muscle in this area as well. Denote that as SM for smooth muscle, or SMU maybe. And then, of course, we're also seeing the pseudostratified ciliated columnar cells as well. So there's our epithelial layer. And some of these other layers that we talked about before, maybe a little bit too much into that area because actually getting a little bit into that smooth muscle, but that's the LP. That'll be our wonderful lamina propria there. Next, we're going to look at an actual bronchiole. So one of the major differences that we can see here is the fact that the bronchiole does not have the hyaline cartilage. So hyaline cartilage is absent. So I'm just going to do a negative hyaline to denote the fact that you don't see hyaline cartilage in the bronchiole. It's lacking. There is, however, lots of smooth muscle. So we still see smooth muscle in this area. So you can see there's some smooth muscle associated with the bronchi. And of course, we still have a respiratory epithelial layer closest to the lumen. So in this case, whether we're talking about the terminal bronchiole or the respiratory bronchiole, for the terminal bronchiole, it's not going to be pseudostratified ciliated columnar cells, but you will see columnar cells typically in this epithelial layer of the terminal bronchiole. For the respiratory bronchiole, it typically is cuboidal epithelial cells that line that epithelia of that mucosa of the terminal bronchiole. Of the respiratory bronchiole. All right. Other structures are still there, obviously, so you still have the lamina propria. There's still, obviously, a submucosa in this layer as well. Here we're actually, I'm going to move to this one. Now we're getting even further down in terms of microscopy, down to the smaller divisions of the lung, even the smallest division of the lungs, which are going to be the individual alveoli and some of these structures associated with them. So the first thing I want to point out is actually the alveoli themselves. So all these kind of octagonal, hexagonal, pentagonal, these kind of shaped rings, if you will, that we see all over this image are all basically alveoli. And some of them are open, some of them are kind of completely enclosed, but these are all alveoli. Okay, a lot of what we're seeing, obviously, if you can envision, this is a three-dimensional structure, but we're seeing it two-dimensionally, so a lot of things get cut off, smushed, mashed, etc. So it may not exactly look like how you would expect it to look in a three-dimensional model or in three-dimensional view, but at least we can see a lot of these structures. The other thing I want to point out too is the alveolar sac. So this is a really nice one right here. So we can see here all the alveoli kind of lining this more open space right here. So all these alveoli together with this shared kind of open space, this would be an alveolar sac, and I'm going to denote that with just an AS. The last thing I want to point out here that we can see, there's quite a few of them, some of them are definitely more definitive, are the alveolar ducts. So for example, this structure right here, this little passageway channel that opens up into some of these alveolar sacs, that's going to be the alveolar duct. So I'm going to denote that with an AD. All right, so if you can envision the terminal bronchiole basically continue on as the alveolar duct, and then the alveolar duct basically kind of open up into these alveolar sac, and then you have lots of these alveoli kind of surrounding the alveolar sac. It's worth noting that the terminal bronchiole have kind of sparse alveoli connected with it, and of course the alveolar duct, as we can actually see here, have some alveoli connecting on the alveolar duct itself. But then you see the density kind of really with the alveolar sac. So you see a lot of these alveoli kind of surrounding the alveolar sac. And for me particularly, I kind of often associate that as like grapes. So So this is like a grape cluster kind of off that vine, and that vine being that terminal bronchiole and that alveolar ducts that basically feed into these clusters of grapes. Okay, the last one we're going to talk about here. So this could be part of a terminal bronchiole coming in, but definitely this would be the alveolar duct in my opinion. So I'm going to denote that with the AD. There is some cellular stuff in here, but it doesn't look like actual red blood cells. So to me, I think this is just some of the tissue and some of the cellular stuff that got stained. So it kind of looks like it's filling these spaces. And indeed, it kind of, obviously, it being three-dimensional, is kind of filling these spaces. But it doesn't look like red blood cells, so I definitely don't think we're looking at a blood vessel. We would see some other structures that would definitely stand out, aside from red blood cells in there. Okay, so we have two types of cells to talk about regarding the alveolar cell. So there's type 1, which I'm going to denote with the Roman numeral I. So type 1 are the majority of these cells that are especially involved with gas exchange. So type 1 alveolar cells are the major cells that make up the alveolar epithelia. All right, the other one are type 2. So type 2 alveoli are producing the surfactant, which helps to reduce the fluid in the lungs, so it reduces the hydrogen bonds by reducing surface tension. So therefore, it's very important to maintain this particular layer for gas exchange. There is some differences in terms of the cell types that we're talking about here. So these are both epithelial cells. Type 1 tend to be squamous cells. So those flat, kind of irregular shaped cells. And type 2 are more cuboidal, more cuboidal in nature. All right, so this is something to think about when you're looking for these in an image. So right off the bat, some of these really look kind of more predominant. So this right here, a little bit cuboidal-like, as is this one right here. You can even see some of these. Definitely that looks more cuboidal shaped, and some of these look a little bit more round, but these would be type 2. So these are those pneumocytes or those alveolar cells, those type 2 that are producing the surfactant. So that's their primary role. For the type 1, these are squamous, so especially where you see dense areas that are thin, that are smushed, they will be most likely type 1. So these are definitely type 1. These are squamous cells, so they're going to be more flat, irregular shape, and you're going to see a lot of them kind of embedded throughout this. All right, so it's best to try to find that one thing that represents that cell type, and that will be much easier when you go to actually try to identify these. The last thing to talk about too would be a roaming macrophage, and I do believe we actually have a pretty nice one here. Definitely have a stained nucleus, albeit it's not typically large like you expect with macrophages, but it is a very large mass. It could just be the way this was essentially preserved and laid down on the actual slide, but it's a very large, looks like a very large plasma membrane with a nucleus in the middle there. So I'm going to call that a roaming macrophage. So roaming macrophages, these are obviously very large cells. Their whole purpose is phagocytosis, so they're going to be very large so that they can actually phagocytize things. All right, so I'm just going to denote that with RM. Okay, so hopefully you found this useful. I'm going to do more histology slides because just from my experience of teaching anatomy and physiology, there are many things that students obviously struggle with, but one of them being histology. So I will make more videos